Hello everyone, can you hear me? I hope you can hear me. Welcome to the week 1 question answer session of the NPTEL course Fundamentals of Protein Chemistry, coordinated by Professor Das Gupta from the Department of Chemistry, IIT Kharagpur. I am Bias Mukherjee, a senior research fellow from the Department of Bioscience and Biotechnology, IIT Kharagpur. And in this session, I will help you with the lecture, week 1 lecture of this NPTEL course. So we will go through the different topics of amino acids. We will see how amino acids are important in our day-to-day lives, and you can ask me any questions that you have during the session or after too. Okay, so we will be beginning in some time from now. Let the others join too, and then I'll give you a brief description of the course, and then we will start with today's class. Okay, so let's give you an overview of what we are going to learn today. I'm sure you've attended the lectures of Professor Das Gupta, and in this session, we will actually discuss the assignment questions. Okay, there are new joinees and methyl. Yeah, so in this course, we will see what amino acids are, and then we will look at the essential and non-essential amino acids, the biological roles of amino acids, and then we will go into the different properties of amino acids. Okay, we will learn about pKa, pKr of amino acids, isoelectric point, and then we will see the stereochemistry of amino acids. We will look at the hydropathy index of amino acids, and we will solve related questions. Then we will look at the titration curve and properties of the peptide bond. Okay. So please ask me questions. If you're not understanding something, I would be really glad to help you out. And I hope this is going to be a learning experience for all of us, for me as well as for you all. Okay, if I'm not able to answer your questions today, I'll come back in the next week and I'll definitely try to answer your questions. Okay, we'll discuss the previous year's questions of the assignments. These assignment questions will help you to solve your upcoming assignments of this particular year. Okay, and this is eventually going to help you to perform well in the competitive exams and also your this NPTEL examination. Okay, now this is going to be a 2 hours session, and to stop you from getting bored, we will be covering few topics and then we will be looking at the questions from those particular topics. Okay, and then again we'll go for a different topic and then look at the questions. This way hopefully you will not get bored. So let's start. All right. Right. So let's talk about amino acids first. Now, what are amino acids? Amino acids, they are the building blocks of proteins. Proteins, I'm sure you all know, we see them everywhere. We are made up of proteins, right? So, how are these proteins made of? So, they are made up of particular kind of molecule which is termed as amino acids. Now, why is this name given? You see, this is how the molecule looks. So, there is a C carbon which is called the alpha carbon, and to the alpha carbon this carboxylic group is attached and the amino group is also attached, and therefore this is called an amino acid. So this carboxylic group, one second. Yes, if you have anything to say you can also write in the chat box. Okay, so let's continue. Right. So we were talking about amino acids, and amino acids, so they are having this amino group, they are having the carboxylic group, and they also have a side chain. So this side chain can be different chemical groups, and these actually distinguish every amino acid from each other. And the fourth one is this hydrogen atom. So this is the basic backbone of an amino acid, and the side chain gives it different identities. Okay, so now this, since this is an acidic group, I am sure you understand that this is ionizable. So this can actually deprotonate. It can give out this H as H plus ion in solution, and it can acquire a negative charge based on the Bronsted-Lowry concept. Okay, and similarly, this amino group at a specific pH, this is going to accept H plus ion and it is going to gain a positive charge. Okay, and therefore amino acids are positively charged. Now, because of this charged nature of amino acids, the proteins are folded in different ways. The different residues in proteins interact differently, and because of that, the proteins are having different functions, different structures, and this entire biological world exists because of all of these properties of its basic molecules. Okay, so now let us, I'll actually increase this full screen mode. Okay, I hope this will be easier for you to understand. Right. So now see, amino acids are amphoteric in nature. So what does that mean? That means that amino acids are having both positively charged basic group and a negatively charged acidic group, and they are having ionizable groups, as I told you. Okay, so therefore they can act as both acid as well as base, and that is why they are called amphoteric. Now, amino acids can be standard amino acids or proteogenic amino acids, and they can also be non-standard amino acids. Now, what are the standard amino acids? Standard amino acids, they are basically coded by genes. Okay, so from DNA, RNA is formed, and from RNA we get proteins. So this is how, based on the codon, your tRNA comes and translation happens, and this is how proteogenic amino acids, standard amino acids are made, and these amino acids result in the formation of proteins. Okay, so they are genetically coded, and there are total 20 common amino acids, and 2 very special amino acids are there. For example, selenocysteine and pyrrolysine. So pyrrolysine is found in bacteria and archaea, and selenocysteine is nothing but cysteine with a selenium group instead of sulfur. Okay, now what are the non-standard amino acids? So non-standard amino acids are not involved directly in protein formation, but they are involved in different physiological processes. For example, the citrulline and ornithine, they are important in the urea cycle. GABA, this is gamma aminobutyric acid. This is an important neurotransmitter in the human brain. Okay, then of course you might have heard about your hydrolysine, hydroxylysine, and these modified modified amino acids which are found in collagen. Right. So amino acids are involved in different essential life processes, including building and repairing of tissues, enzyme activity, digestion, assimilation of food, signal transmission, sensory perception, and so on. So now, what are essential and non-essential amino acids? Now, as we are eating, certain amino acids can be synthesized by our body. Now, these are called these are called essential amino acids. Right, sorry. So out of the 20 amino acids, our body can synthesize some amino acids, and these are called the non-essential amino acid. The rest of the amino acid is introduced into our body through diet, and these are called the essential amino acids because the body cannot synthesize it by itself. Okay, so essential amino acids are these isoleucine, leucine, lysine. These are all essential amino acids because these need to be introduced into our body through our diet. Now, non-essential amino acids, they can be through different metabolic processes. Our body can actually synthesize this. Right. So there are 2 amino acids which are semi-essential. That means that they are produced by our body but not in sufficient quantities. So arginine and histidine are the amino acids which are semi-essential, and therefore a portion of this needs to be provided through our diet as well. Now we will look at the biological role of the amino acids. Okay. Right. So, breakdown of food, growth and repair of the body tissues, hormone production, and making the brain chemicals. Besides that, providing energy, and then maintaining healthy skin, hair, nails, build muscles, boosting the immune system, and sustain a normal digestive system. So these are the wide variety of functions that amino acids perform in our body, and this tells us how important amino acids are. So now we come to an important question. Now this tells you how important amino acids are. So then can you tell me whether we should actually consume amino acid supplements or not? Because these days protein supplements, they are very common. Everybody is taking protein supplements and all, right? But then do you think this is actually necessary, or can we do without consuming amino acid supplements separately? What do you think? You can answer in the chat box. Okay, you can see the answer, I think. I'll just go to a different slide. Yeah, now you can answer me. You can just type in the chat box. What do you think? Amino acid supplements must be given? Yeah, please tell me. Yes, someone raised your hand. You can say, you can answer. You have anything to say? Okay, so let's try to find the answer. So in today's age, you might be thinking that yes, this might be very essential to actually consume supplements and all of amino acids. But no, the right answer, science says, is that through our food, we can get all the necessary dietary supplements that we need. Okay, and that comes from food. You do not need to take additional supplements to have a healthy body. But then yes, sometimes there is deficiency, let's say of vitamin B12, of biotin. In such cases, if the doctor prescribes, additionally taking these amino acids can really help to improve your health. Okay, so see, deficiency of amino acids actually can cause headache, weakness and irritability, fatigue, skin and hair related issues, insomnia, depression, loss of appetite, diarrhea and anemia. So these are the varied conditions that the body experiences in deficiency of amino acids. Right, so now we will move on to the properties of amino acids. Now this is a very beautiful chart which gives you a very vivid idea of the different types of amino acids that we have. So all these classification of amino acid, this has been done based on the R side chain. Okay, so I told you that R side chain is like your name, it gives you a definite identity. Right, so similarly, if you look here, hydroxylic chemical group, right? So that means there is serine and threonine. So if you look at the right hand side, see this S and T, this is serine and threonine, and the single letter code is given, and this is the name of the amino acid, and then the 3-letter code is given. So you must remember all the names. Okay, and so serine and threonine both have hydroxylic group in them, and therefore they are hydroxylic amino acids. Now beside that, there are aliphatic amino acids. Aliphatic amino acids have this hydrogen and carbon chain. There is sulfur containing amino acids such as methionine and cysteine. So based on the properties of the amino acids are classified into various types. So there are non-polar side chain amino acids which have non-polar side chain. So they do not participate in intermolecular hydrogen bonding. Okay, so for example, alanine, but this participates in your hydrophobic interactions. Okay, so there is alanine, there is leucine, isoleucine. They are having non-polar side chain, but they participate in hydrophobic interactions. Okay, then we have the polar uncharged side chains. So amino acids which have, yes, okay, so we are welcoming a lot of new members. That is very good. So polar uncharged side chain, the amino acids that have non-ionizable side chains and participate in intermolecular hydrogen bonding or ionic interaction. So for example, serine. Now serine is having one electronegative atom which is O, and another atom which is H. Now this oxygen, this has a very strong electronegativity, and therefore it develops its delta minus charge on itself, and H has delta plus charge. But yeah, ma'am, will we get the PPT after the lecture? Yeah, definitely you'll get the PPT. Okay, you'll have the PPT, and you will also have the recording of the lecture on YouTube, uploaded on YouTube. So all of that will be shared by NPTEL. Okay, so that does not mean you'll run away now. Try to attend the class and try to ask questions if you have any doubts. Okay, so yeah, so what does it mean by non-ionizable? So basically this serine OH, this is not going to break into H plus and OH minus, right? This is just going to be participating in the interactions, but then it is not going to break, it is not going to be ionizable. So the last one is charged side chain. So charged side chain has positively charged NH3 plus, for example seen in lysine, it is seen in arginine, and negatively charged R side chain, that is seen in glutamic acid and your aspartic acid. Okay, so this chart I hope will be very useful for you to remember the properties of these amino acids. Now these are the amino acids at a glance. So I remember it was very difficult for me also to remember the structures of the amino acids, but then sometimes for competitive exams structures are very important, and so the way to remember them easily is to actually understand them. Okay, so there is this hierarchy. So if you look at glycine, this is the simplest amino acid and it is having hydrogen, and it is having two hydrogen groups attached to this alpha carbon. Okay, then remove one hydrogen and attach a methyl group, that makes it alanine. Then what happens? You add another methyl group, and this is the T-shaped structure that you're getting. This is actually valine. Then you just increase one methyl group here, CH2 group. You just increase one and you get leucine. Similarly, you get isoleucine, and methionine has this sulfur in it, right? Then there is phenylalanine. So phenylalanine is having alanine with a benzene ring attached to it. That makes it phenylalanine. Now this is an aromatic amino acid. Why? Because this is having this conjugate double bond structure, and so this is an aromatic amino acid. Then there is proline. Now proline is the most unique amino acid. Why? Because here the alpha carbon, it is attached to this NH group, and it is also attached to another CH2 group, and there is a ring structure here. Okay, 5-membered ring. So because of this steric hindrance, proline is not very easily seen in alpha helices, and it is called a helix breaker. So this structure of proline gives it a lot of different properties. We will learn about that eventually in the next classes. Okay, and then there is tryptophan. So tryptophan has this indole ring. Now see here, because of this conjugate double bond structure and delocalization of electrons, this actually absorbs a lot of light. And so if there is tryptophan in your amino acid, in your protein, you actually get a lot of absorbance through absorbance spectroscopy, and it is very easy to quantify that protein if tryptophan is present. Otherwise, it becomes very difficult to quantify the protein. Okay, and then there is asparagine. So asparagine is having this CONH2 group, and then there is glutamine. Glutamine also has CONH2 group and extra one CH2 group than asparagine. Then there is serine. Serine is actually this alanine, and you're having this OH group attached. Okay, then there is threonine. Threonine just has one additional methyl group compared to serine. This is how you can remember the amino acid structures. So let us start with the first set of questions. So you try to answer the questions. Okay, you try to write down the answers in the chat box, and then we will discuss. Okay, so the first question is, which of the following sets contain all polar neutral amino acids? So who are the polar neutral amino acids? Find out from the options. Okay, so the first option is glycine, aspartic acid, and your asparagine and glutamic acid. Then the second set of options are all the aromatic amino acids. The third set of options are serine, threonine and cysteine. Okay, what does cysteine have? Cysteine also has sulfur, right? And then there is isoleucine, valine and leucine and methionine. You try to give me the answer of this question. Write down in the chat box. For the next question, it is the following sulfur containing amino acids. So I actually gave you the answer to this question. Okay. So try to answer the questions in the chat box. B. Okay. So B is the answer for which question? First one or the second one? Cysteine and methionine. Yes. Cysteine and methionine is correct. Okay. I think they have given this, acha. Cysteine, the spelling also you actually need to note, okay, which is the exact spelling of cysteine, because see, there are two different molecules given. One is cysteine and the other one is cysteine. These also you need to note. Okay, so let us look at the answers. So, this is the right answer is C. C has serine, threonine and cysteine. Why is that so? Let us look at this chart. Okay, so right, we can actually have a look from here. So this is threonine, this is your serine, and this is cysteine. So all of these have this hydroxyl or sulfuryl group. Right, so sorry, thiol group, this is SH group and this is OH group, and because of this it is polar neutral amino acid. They are polar but then they are neutral. Okay, and the second question, the answer to the second question would be methionine and cysteine, because both of these contain sulfur. See here, methionine and cysteine. Okay, so we move on to the next topic, which is stereochemistry of amino acids. Okay, so right, now stereochemistry of amino acids. So if you look at glycine, what is exactly chirality? I'm sure a lot of you already know. So chiral carbon is a carbon to which 4 different chemical groups are attached. Okay, so if you look at glycine, glycine is not a chiral carbon because this has 2 hydrogen groups attached to the carbon. But then if you look at alanine, this is having 4 different chemical groups attached, and therefore we can call it chiral. Now because of this chirality of amino acids, they have a property of interacting with light, and when plane polarized light strikes the amino acid, this actually, because of the chirality of the amino acid, this changes the polarity of the light, and so the light that we obtain after it has passed through the amino acid solution, this is no more plane polarized. Okay, so a lot of different experiments have utilized this. For example, there is circular dichroism spectroscopy, which has utilized the property of amino acids to actually interact with circularly polarized light. So now we have L-alanine and D-alanine. So what is this capital L and capital D? A lot of people have confusion regarding this. So if you look at this particular representation, Fisher projection formula, if you see, sorry. Right. So L and D amino acid means that the absolute configuration of a molecule that predates R and S. So R and S are again a different way of naming the molecules. So see, for this molecule, this is L-alanine. So here in L-alanine, this amino group, this is, if you look at your left hand, this amino group is actually pointing towards the left, and this is actually, it is not the left, this is actually pointing towards your towards the inner side of your, towards your side of the body, right, towards the inner side. And this is the D form, where this NH2 group is actually pointing towards the other side. And so this is just how you write it. So for example, this L NH2 group, this is written towards the left, and this D NH2 group is written towards the right. So when the amino acid NH2 stays on the left side of the amino acid, then it looks like an L-shaped amino acid, and this can be thought of as left hand. Right, this can be thought of as a left hand. Similarly, when the D-shaped amino acids are just mirror images of the L-shaped amino acid, and so this can be thought of as the right hand. Now what about this dextrorotatory and levorotatory? So when light, through a light source, you know that light is made up of electric field and magnetic field vectors, and these actually oscillate along all directions. And then when we pass this light through a polarizer, you can imagine polarizer like a slit, and this allows the light of having only vibrations along a particular direction to be passing through. So the electric field vectors vibrating only along this particular direction passes through, and this is now called plane polarized light. Now when this plane polarized light comes and hits the amino acid sample, then they actually bend the plane polarized light, and you can see that this is now in a different plane. So this has been rotated, right? So this is how amino acids interact with plane polarized light. Now the amino acid which are dextrorotatory or D amino acid, small d. So they actually rotate this plane polarized light towards the right by some degree, and the other one, which is your levorotatory, they rotate the plane polarized light towards the left by some degree. Now you tell me, if they are present in a mixture, then what will happen? Some of the molecules will rotate the light towards the right, some will rotate it towards the left, and then there will be net rotation would be negligible. Can you send the videos which you were talking? Sorry, can you send the videos which you were taking a class? Right, yeah, of course. You'll see the videos on YouTube. All these videos will be uploaded. Okay, so even if you miss any class, there is nothing to worry about. You can always go back to the videos. You can always go back to the assignments, assignment questions, go through them, go through the solutions. Okay. All right. So let's continue with the session. Right. So this is all about D-formed dextrorotatory and levorotatory forms. They can be present as a racemic mixture. Now, in nature, majority of the amino acids are L amino acids. Okay, capital L. They are majority capital L amino acids. Does that tell us that there are no D amino acids? No, that is not the case. If you remember, in the cell wall of bacteria, it is made up of this NAG and NAM, right? N-acetyl muramic acid and N-acetyl, right? So N-acetyl muramic acid and N-acetyl glucosamine. So here, if you see, there are D-amino acids. They are present as the pentapeptide. This is where your penicillin goes and binds and attacks, right? Prevents the formation of the bacterial cell wall. This is how it actually kills the bacteria. So, D-amino acids like D-glutamate and D-alanine are actually found in nature, and they are seen in these bacterial cell walls. Now we come to the questions. Okay, you try to answer the questions. They have asked you about the chirality of amino acids. So, isoleucine, serine, tryptophan and threonine. Which amongst these do you think are most chiral? Okay, which is having the most number of chiral centers? Tell me. You can answer in the chat box. Okay. So if you remember the structure of isoleucine, it is having a very long aliphatic chain as the R group. What about serine? It is just having OH group. Right. Then there is tryptophan. Tryptophan is having this indole ring. And then there is threonine. So threonine also has a very long chain. So you tell me the answer then. I have given you the clue already. Threonine, yes, very good. So see, in none of the options there is only 4, right? So there might be one more amino acid with threonine. Which one do you think? C. C is 1 and 2. Okay. Let us look at the right answer. So right answer is actually D, 1 and 4. So see, isoleucine, this is the structure of isoleucine. This is one chiral center, this is another chiral center. So, and about threonine, this is one chiral center and this is another chiral center. So both of these amino acids actually have 2 chiral centers. Now most amino acids have only one chiral center, but this is a rare case scenario where an amino acid is having 2 chiral centers, and therefore these are very, very special. Okay, these are simple questions. We'll go to the complex questions very soon. Now we come to hydropathy index. So what happens in hydropathy index? Now this is actually a measure of the hydrophobicity and hydrophilicity of amino acids. Okay, and this is used to predict the behavior of amino acids in a protein, particularly how they will interact with water and how they will be distributed in the protein's three-dimensional structure. Okay, so if a particular amino acid is actually hydrophobic, it will fold towards the interior of the protein, right? The protein chain will be such that it will go towards the interior. But if the amino acid is actually polar in nature, if it is charged in nature, then it will be towards the surface of the protein. Mostly it will interact with the water molecules, and it will also interact with ligands and every other entity, right? So therefore, if you look at this plot, this actually is a plot of hydropathic index. So the hydrophobicity of the amino acids are given to the residues which are having positive hydropathic index. So they are actually found on the surface of the proteins. But then, okay, hydrophobicity, yeah, they are found in the interior of the protein. But then the residues which are having negative hydropathy score, they are found in the surface of the protein. For example, if you see arginine is having a very high hydropathy index of minus 4.5. So negative but very high, right? So this is actually very much hydrophilic. Similarly, your glutamic acid is very hydrophilic, and so these hydropathy index can tell you about the presence of the amino acids in a protein. For example, if you look at this transmembrane protein, there is a high probability that leucine, isoleucine, this will be present here in the transmembrane domain, right? But then this arginine, this might be present here in the cytosol, where it will be interacting with other water molecules and other ions. So now try to solve this question on hydropathy index, and you ask me, stop me and ask me if you're not understanding something, right? So try to solve this question. Identify the amino acid with the lowest value for the hydropathy index. C, arginine. Very good. Yes. So it is negative and it is the least, right? So arginine is definitely the least. This is mostly present on the surface of the protein, right? See, as I was telling you, isoleucine, valine, these are mainly present in the membrane spanning regions of the proteins because they are highly hydrophobic. Okay. Now we move on to the isoelectric point of an amino acid. So, at the physiological pH means the body pH, which is actually different regions in the body have different pH, right? Inside the cell also different compartments are having different pH, but then we take the physiological pH as around 7.5. So, around 7, let's say. So at the physiological pH, most amino acids exist as zwitterions. Now what is this zwitterion? Zwitterion is actually having both positive and negative charges. Okay, the pH at which the amino acid has equal number of positive and negative charges such that the net charge is 0. That is called the isoelectric point of the amino acid, and the amino acid is said to be zwitterion. So the pH values above and below the pi, the amino acid is actually positive and negatively charges respectively. It is having positive and negative charges respectively. Okay. Now, a typical amino acid with neutral R group transitions from positively charged to a zwitterion and then to a negatively charged. Now, we will learn about this very soon, why exactly this is happening. So, at a very, very strong pH, okay, very strong pH, this COOH group exists as COOH instead of COO minus, okay. But then this NH3 groups, this remains protonated. Therefore, there is +1 charge. Then as you move to physiological pH, what happens? The pKa of this particular acidic group is reached, and this gives out this proton and it becomes negatively charged carboxylic group, right? And then there is your net charge becomes 0. Then the finally, as we increase the pH, what happens? There is further deprotonation and this NH2 group actually loses H plus ion and we get negative charge only. Okay. So the zwitterion is actually a form of salt. and it is formed as a result of acid-base reactions of the basic and the amino group and the acidic carboxylic group. Now, acid-base reactions are the following: deprotonation or loss of H plus ion of the COOH group as the pH increases, and then there is also protonation of the H plus ion of the NH2 group as the pH decreases. So if we move from here to here, there is protonation. If we move from here to here, there is deprotonation. Okay. Now, how is the isoelectric point of an amino acid determined? Right. So for this, there is the Henderson-Hasselbalch equation. Henderson-Hasselbalch equation tells you that pH is equal to pKa. Now this Ka is actually dissociation constant. Okay, dissociation constant of an acid. Now this P, of course, you know, this is negative logarithm to the H plus ion concentration is equal to the negative logarithm to the dissociation constant. Then the concentration of the H plus ion and concentration of the conjugate base is exactly the same, right? So log of these two, log of 1 gives you 0, and therefore pH is equal to pKa when these are in equimolar concentration. Excuse me. Right. So see the quantitative measure of the strength of an acid. This pKa is actually the quantitative measure of the strength of an acid. When the concentration of acid or HA is the same as that of its conjugate base, which is A minus. So lower pKa values means the acid is stronger, and the higher pKa value means that the acid is weaker. So pKa is the pH at the midpoint of the flat buffering zones in a titration plot. So if you look at this, actually this is the region of the neutral charge of the amino acid, and here is when the amino acid is positively charged, and here the pH is low, and here the amino acid is negatively charged. So at this midpoint, here is when you get to see the pKa1 and pKa2 of amino acids. Now beside pKa1 and pKa2, for amino acids with charged side chain, there is a third component which is pKr for the R side chain, right? So the isoelectric Pi lies between the points A and B, where the concentration of an acid is same as that of its conjugate base. Fine. Now we will.
Yes, ma'am.
Yes.
I think slide is not changed.
Slide is not changing. Okay, I'm so sorry. Okay.
When
Which slide are you in right now?
now we are seeing it 19th slide, but we haven't seen the 18th slide when you are explaining it.
Achha.
We just seen the 17th slide.
Okay, okay. Then I'll not share the screen then, okay? It delays the entire process. We'll do it this way. So this is the 18th slide, and in the 18th slide you see that this A and B are the regions where your pKa1 and pKa2 come, and this is the Pi, and this is how from as the pH increases, the amino acid transitions from positively charged molecule to neutral molecule to negatively charged. Okay, we will look at the example after this to exactly understand what happens. So let's see the example. So firstly, there is this in the first one is for an acidic amino acid. So see pKa1, okay, for that is 3.65. Now pKa1 is always for your alpha carboxylic acid. Okay, pKa1 is always for alpha carboxylic acid. pKr in this case is your 1.88, which is for the R side chain. So what happens initially when the pH is, let's say, 1, then the amino acid is positively charged. Why? Because this COOH, both the COOH are actually present as COOH, but then this NH3 is present as NH3 plus. So it is positively charged. Then what happens at pH 1.88? This R side chain, it actually leaves one H plus ion from COO group. So then what happens? That positive charge was there and we get one negative charge. So we get neutral charge here. Right. Then after that, as we move to pH 3.65, then what happens? Another H plus ion is gone. So it is net negative. Okay. So Then these amino acids, this negatively charged amino acid, they have very low pi value. Similarly, for neutral amino acid, I'm sure you've understood already. And for the basic amino acid, very easily, this COOH group is actually leaving the H plus sign when the pH is 2.18. Beyond 2.18, it is actually getting deprotonated. Then what happens? Both the plus 1 charges remain and the pi is reached only when we reach the pH of more than 8.95. So around pH 9 or something, this H plus sign is lost, and then we have net neutral charge. Okay, so to calculate the pi of a neutral or diprotic amino acid, diprotic meaning that there are two different chemical groups, right? An average of the pK1 and pK2 are taken. However, to find the pi of the triprotic amino acid, the average of the pKa values that are closest to the pH at which the amino acid assumes net zero charge is actually used. So, for example, for the positively charged amino acid, for the so for the positively charged amino acid, which is lysine, which two will you take? The ones that are closest to net neutral charge. So this 8.95 and 10.53, and therefore your pi is going to be very high. Similarly, for the acidic amino acid, your pi will be very low, and you will take the two pK values that will give you, that are very close to the point where the net charge will be 0. So pK1 and pKr. Okay. Now we'll go to the next one. You tell me if you're not understanding. Okay, so this is actually a list of all the pK values of the different amino acids. You can just go through this and have a look. Okay, then these are the charged amino acid. You can see that every chemical group, all the ionizable group, they are having their different pK values. Then this is a chart which tells you about the different amino acids, the different side chains that they have, and the pK1 of the alpha carboxylic group, the pK2 of the alpha amino group, and the pKr of the R side chain. So these are the isoelectric points of the amino acid, and from the isoelectric point you can actually tell whether the amino acid is neutral or it is your positively charged or negatively charged at neutral pH. Okay, so neutral pH, you see this is around 6 to 8, like that. So all of these are, this is how you can actually tell about the properties of the amino acids. Right, now try to solve these two questions. Question on the pi of amino acids. Now questions on pi are going to be there for every exam, and so you have to practice these questions very well. You have to be very well versed with these questions. These are very scoring, very simple, but very confusing. They'll give you the values so close by that you will be very confused. Okay, so try to solve them. These are very simple questions here. So an amino acid with neutral side chain has a pK1 of 3.4 and pK2 of 9.4. So pK1, it is a neutral side chain, right? So very simple. At which value of pH will the amino acid migrate towards the anode? So first question is the anode positive or is it negative? First you answer that. Anode is positively charged or anode is negatively charged? During electrophoresis, if the amino acid is charged, it is always going to migrate to the charge which is having oppositely charged electrode, right? Very good. Yes, anode is positively charged. So that means we want to know which amino acid is going to be negatively charged at, right? So when will it be negatively charged? So at which pH value will it be negatively charged? So for that, what do you have to do? Okay, positive 6.0. Okay. And for the second question, we look at this answer very soon. Right. Acha. So two things you need to keep in mind. Firstly, so yes, that means the amino acid should be negatively charged. That is point number one. Second point is at which pH? Below the pi or above the pi? Should it be that it will have negative value? So that is the second thing that you need to keep in mind. Pi, I am sure you've all calculated, right? Pi you definitely must have calculated by now. Now at what pH? Should it be below the pi or should it be above the pi? Such that it will be your negatively charged. Okay, we will see. Two amino acids having R groups with a negative net charge at aspartate and glutamate. Very good. Okay, so we will now look at the answers. Right. So, yes, this is definitely correct. Aspartate and glutamate. Very good. For this one, you see, this is your pi is actually 6.4. Now, if you look at this figure, below the pi, it is positively charged, right? Because, and above the pi, okay, 1 second. Right, we will just look at this answer. So only at pH above 6.4, the amino acid will be negatively charged and migrate towards the anode. Right? Here, the negative form will be dominating. See, this is positive and this is negative. As the pH increases, the net charge is negative. So that means it is going to be 7.4. That should be your right answer. Right? Now we will go to the next question. I hope it is clear. Okay. Now you try to solve this. Right. So this is your titration curve. Now what happens here is there is a particular amino acid given, which is your arginine. Right. So now this arginine amino acid, this is a titration curve for arginine. Okay. So initially there is pKa1 at which the COOH group is becoming COO minus. Then there is pKa2. So pKa2 is actually for the alpha amino group and your pKa3 it is for the R. Okay, pKr. So the pi is definitely between this alpha amino group pKa and the R side chain pKa. So now they are asking is which of the following ionic structures would be expected at pH 8. Okay, so pH 8 is this. So which charge will it have at this particular pH? That they are asking you to find out. Tell me, think and tell. At this pH, what is going to happen? Excuse me. At this pH, at a pH of less than 2, this should be the structure, right? But after 2, these 3 should be the structure, A, C or D. So it is either A, C or D. Now, now what happens? This alpha amino group loses the H plus ion when the pH is more than 9. So below 9, what should the case be? What should the scenario be? Tell me. Acha, you're saying it's A. Okay, let us actually look at the answer. So the answer is not A. Why is it not A? Because at a pH of greater than 2, already this is the form, right? More than 2, this is the form. Then what happens? Then at a pH of 9, this particular, right, 1 second. So less than 2, this is the form, but just beyond 2, D is the form. Here what happens, this alpha amino group exists as NH3 plus and the R side chain exists as your NH3 plus. Okay. But then this COO group exists as COO minus instead of COOH. So that is the right answer. I am sure you've understood. If you have not understood, please stop me and question me. Okay. Now we will look at the next question. So the Henderson-Hasselbalch equation. So what about the Henderson-Hasselbalch equation? Try to answer this. So Henderson-Hasselbalch equation does not explain the behavior of di or tribasic weak acids. It is equally useful with solution of acetic acid and hydrochloric acid. Relates the pH of a solution to pKa and the concentration of acid and conjugate base. This should definitely be correct. Allows the graphic determination of the molecular weight of weak acid from its pH alone. So which one do you think would be the right answer? One and three. Okay. So you're saying it's A. Okay. Alright. We will take a look at this later. Let us take a small break from for now, and we will come back with the answer of this question. Okay, let us take a small 5 minutes break and we will be back soon. All right, we'll be back sharp at 7, and then we have a few more slides to complete. We have a lot of questions. So beside your assignment questions, there are also many new questions that I've added. Okay, so it is mostly the new questions, and the only remaining topic is your peptide bond and Ramachandran plot, which are also very, very important. And also we have some portion of bathochromic and hypsochromic shift, which are also very important for your exams. So we will be back after a tiny break. Okay, welcome back. And yes, you're very correct. The right answer is 1 and 3 both. Okay, Henderson-Hasselbalch equation does not give very appropriate answers for your di and tribasic weak acids. So yes, this is the correct answer. Okay, now we move on to the next question, which is a new question. One second. Okay, I think the this is already given. Sorry. Okay. Try not to see now. Okay, and try to answer. Now you try to answer this question. So the pKa2, that is pKa for the side chain of histidine is 6.0 when it is present in an isolated state. Considering a specific protein with histidine at its active site, the pKa2 is expected to be, okay, pKa2 here is actually the pKa for the side chain of the histidine. Okay, so now they are asking you whether this is going to vary when it is present in the active site of the protein in the proximity of other amino acids, right? So when it is alone, it is 6, but when it is present in the proximity of other amino acids, let's see what happens. So less than 6 if the active site has more positive charges. Greater than 6 if the active site has more positive charges. Less than 6 if the active site has more what negative charges, and same like isolated histidine. Now this is a very complicated question. Try to think and answer. Diagram is given for your ease. What happens to the PI? What happens to the pKar of a particular protein when it is present in the vicinity of a particular amino acid when it is present in the vicinity of other amino acids in the active site. Now histidine is very commonly found in the active site of proteins, right? So what is going to happen? A. Okay. Let's see. Let us look at the answer. That's very good. Yes, A is the right answer. So when histidine is present in the active site having mostly positive charges, the pKa2 becomes lower than 6 because the positive charges in the active site destabilize the positively charged histidine side chain. So a higher concentration of protons, that is lower pH, is required to protonate the histidine side chain, therefore resulting in a lower pKa. So this is a very interesting question, and you can actually try to understand how even other amino acids affect the properties of a particular amino acid. Okay, right. So how do we determine the PI of peptides then? Now this you tell me, you have understood, you have learnt about the PI of individual amino acids. But then for your competitive exams, they will not usually ask you the PI of a specific amino acid, but they will ask you the PI of a specific tripeptide or a dipeptide. Then what will you do? Okay, so in that case, you have to consider the pKa values of all the ionizable groups, and that includes the N-terminal amino group, C-terminal carboxylic group, and all the R side chains, right? Because in between, so there is this peptide bond that is not going to get ionizable. So see here, look at this tripeptide. There is an aspartic acid residue, there is a glycine, and there is a lysine. So there are 3 different ionizable groups. So initially when it is pH 1, none of these have pH 1, right? Wait. So none of these have pKa 1. So it is going to be what? Positively charged, and there are 2 NH2 groups. So it is going to be definitely double positive. Then what happens at a pH of more than 2.2, we have this particular group gets deprotonated. So 1 minus charge gets added, and so this is plus 1 now. Then what happens at a pH of more than 4, right? So that is, I will take this as 7, which is the neutral pH. What happens? This particular group, this carboxylic group, so this particular group gets deprotonated. Then what happens? The net charge is plus 2 here and minus 2 here, and so the net charge is 0. Then what happens? Then at a pH of around 10, which is more than this particular group's pH, this is actually deprotonated, this gets deprotonated, so the net charge is now minus 1, and finally this gets deprotonated, so net charge is then minus 2. So this is what you have to understand, and this is how you can actually calculate. So how will you calculate the pI of the peptide? So you will take the 2 pK values, where 2 pK values closest to the point, closest to the pH at which the amino acid, the tripeptide is going to have net zero charge. Okay, so which of these are the two here? So that is 9.6 and 3.65. So this is the one and this is the one, right? So you'll take these two and then you'll find the pI of the tripeptide. Okay, I hope that is clear. The concept is same for all. Okay, just a little more complex. Now what happens? What is the purpose of amino acids? Now one of the major purpose of amino acids is to actually join together to form proteins. Now after that, proteins fold together. There are the secondary structures of proteins, alpha helices and beta sheets. There are tertiary structures between the different interacting amino acids, and finally different subunits of the tertiary structure come and join together to form the functional quaternary structure, right? So here we will actually concentrate on only this today. Okay, we'll concentrate on how the amino acids join together to form the primary structure of protein, and they do that by forming the peptide bond. So in the peptide bond, what happens? There is the removal of one water molecule and the two amino acids get joined together. Now this peptide bond, you can see that this is a single bond, but then there is a carboxylic group here. So there is actually a resonance going on between these two, and there is a distinctive double bond character due to resonance between the different structures. Right, the carbon and oxygen atoms involved in the bond share electrons in a way that creates a partial double bond character, making the bond very rigid and preventing free rotation. These are very important characteristics of the peptide bond. This rigidity contributes to overall three-dimensional structure of the protein. Alright, this is how the resonance happens. Right, and then finally, this is not the final part though. Anyway, we come to torsion angles. So what are torsion angles? As I told you that, now you know that these moieties can rotate around bonds, right? So if you see this as one plane rotating around another plane, and it is emerging from the C alpha. So C alpha, the region this side to the C alpha rotates, and the region this side to the C alpha rotates, and all of these actually rotate as planes. So rotation around the C alpha C is called psi, and the bond between C alpha N is called phi, and these angles of rotation, they are actually called torsion angles. So they can be from 0 to 180 degree. Besides that, there is also an omega angle, which is actually the rotation about the peptide bond. Okay, so this rotation around the peptide bond, but then it is very limited, and therefore we do not take this into much consideration. Okay, so planarity of the peptide bond restricts omega to 180 degrees in nearly all of the main chain peptide bonds. In rare cases, omega is 0 degrees, which are seen in a cis peptide, right? So which is usually seen in the proline. So what is a cis peptide? Actually, in proteins, it is usually like this: CONH. Again, CO and NH. That is actually a transpeptide, because the CO and NH are directing in two different directions, right? But then if it is CO and NH, then this is called a cis peptide, and cis peptides are very common in prolines. But then for other amino acid, it is majorly transpeptides. Now this torsion angles might initially seem a little confusing, but when you go through books and when you read it properly in theory, you will understand that this is a very simple concept. This is actually just two planes rotating around a particular bond. Okay. Now we will look at Ramachandran plot. So based on the probability of the torsion angles owing to the presence of amino acids, Ramachandran has given certain probabilities that actually tell you about the different types of secondary structures that are present in proteins. For example, this antiparallel beta sheet means one beta strand, again beta strand and beta strand. They are pointing in opposite directions. So they will have a phi and psi angle between this range, right? And then again parallel beta sheet, this is these are usually the values for the parallel beta sheet. Again for right-handed beta sheet, right-handed alpha helix, these are the values. And if you plot a graph based on this, so 0 to 180 plus 180 to minus 180 for both phi and psi, you will see that this is the region where the beta sheets come. This is the region where the right-handed alpha helices are present. This is the region where left-handed alpha helices are present. Now in nature, right-handed alpha helices are most common. Okay, and glycine amino acid, this has the least restriction. It can be present anywhere in the Ramachandran plot, and the amino acids containing proline, they are very, very restricted. Okay, so Ramachandran plot is a commonly used tool to visualize the distribution of phi and psi angles in a protein structure and assess the allowed regions of conformational space for a given amino acid sequence. Okay, now you try to answer the questions on peptide bond. Which statement about the peptide bond is incorrect? It is stronger than an ordinary single bond. It has partial double bond character. It is weaker than an ordinary single bond. So, two options oppose one another. That means definitely one of them should be wrong. Correct? Second one seems to be correct, doesn't it? So, then 1 and 2 are correct. So, then we can say that option A is correct. Right? Yes, option A is definitely correct here. That is 1 and 2. Okay. Now, we'll come to the next question, which is a question on the torsion angles. So, N and C alpha. So, bond between N and C alpha is equal to psi. Is that so? And C alpha and C is equal to psi. So, N C alpha is phi and C alpha C is psi. Peptide bond is omega. That is the right convention. Okay. So, see, peptide bond is omega, N, C alpha is your C alpha and C. This is psi, and C and N. C alpha and N, this is phi, and omega is the one for peptide bond. Okay. Yes, this is wrong because this is phi and phi should be this. NC alpha should be phi. Right. So now we will go to absorption spectroscopy and we will try to understand how amino acids behave in the presence of light. Okay. So interestingly, different substances can absorb or emit light at different wavelengths. Okay. This is the basic principle of spectroscopy. So spectroscopy is a technique used to study the interaction between matter and electromagnetic radiation. It involves analyzing the spectrum of emitted, absorbed or scattered light to gain information about the composition, structure and properties of materials. So light comes from the source, enters into the monochromator, a specific wavelength goes to the sample, and then sample actually emits a different wavelength and it is detected, and then the output is analyzed. So absorption spectroscopy is a biophysical technique to measure the absorption of electromagnetic radiation by a substance as a function of wavelength. The basic principle of absorption spectroscopy and UV visible spectroscopy involves passing light through a sample and measuring the amount of light absorbed at different wavelengths. So the extent of the absorption is related to the concentration of the absorbing species and the characteristics of the molecular electronic structure. So this works on the principle of the Beer-Lambert's law and absorption spectrophotometer. This is how the device works. Okay, now we will look at a very interesting and very important concept, which is the Beer-Lambert's law. So for Beer-Lambert's law, you will get questions at every exam. So please remember the formula for Beer-Lambert's law. Okay, so what does Beer-Lambert's law say? Beer-Lambert's law states that for a given material, sample path length and concentration. So what is the path length? Path length is actually from the source, from after entering into the sample, how much light it is, how much path the light is actually traversing. Okay, that is the sample path length. And the concentration of the sample are directly proportional to the absorbance of light. So this directly equates the concentration of the sample to the amount of light it absorbs. So amount of light it absorbs, that is measurable. That means this is how you can actually measure the concentration of your protein solution. Right. So A is equal to epsilon into, yeah, sure. What is your doubt? You can tell me what your doubt is. So as long as he is saying, let us keep on. Okay. If you're writing in the chat box, you can please write, and then as soon as I see, I'll answer. Okay. So C A is equal to epsilon into C into L. So epsilon is your molar extinction coefficient. Now this depends upon the properties of the protein. Okay. So and the properties of the protein means the number of amino acids that are present which actually absorb light. So mainly the aromatic residues, they are mostly involved in absorbing light. Okay. So the number of aromatic residues present, that gives you this molar extinction coefficient. Okay. Then there is C, which is for concentration, and L is the path length. Okay. Kirti Ashok Kumar, you can actually ask your doubt or you can write. So in class 12th, we studied the same concept of the top of the before topic. Yeah, of course. This is actually, see, basically a lot of this you will have studied because this is just the beginning of the course, right? As we advance, you will study different things. So this is actually, maybe this is a brushing up for you, but to this course actually people from different background come, and so we try to do it in such a way that everyone understands. So I'm sure you understand it very well then if you've already done this in 12th. Okay, so then it will be easier for you to actually solve the questions, right? So yeah, so let us continue. Okay, so C is the concentration of the absorbing species and L is the path length. So as we go to the other lectures, as we move on with the course, you will see that you will find new things, hopefully. Okay. Achha. So what is Beer's law and what is Beer-Lambert's law? So before that, you see this absorbance, this can be seen in different ways. So absorbance can be log of I naught. I naught is the intensity of the incident light and I is the intensity of the emergent light. Okay, so you can see, look at this diagram here. This is the incident beam and this is the emergent beam. Okay, with lower intensity because a lot of light has been absorbed by the sample. Right, so then there is again Beer's law, which states that the concentration and absorbance are directly proportional to each other. Lambert's law states that absorbance and the path length are directly proportional to each other. So if you make the light pass through sample of higher concentration and a longer path length, then more of the light will be absorbed. Okay. Now, this is how an UV visible spectroscopy looks like. Okay. So UV visible spectroscopy, here what happens? Absorption of ultraviolet light. So mainly our amino acids, they absorb light on the ultraviolet and visible light spectra. Okay, this is how the device looks. There is this detector, which is your there is a source of light, which is often tungsten lamp or D2 lamp, and this is going to the mirror. It is coming to the monochromator. There is a single slit, so from there only a specific wavelength is coming. Then there is beam splitter. This is going to the sample. A part of it is going to the reference, because there always needs to be a blank. Blank is just the buffer without the protein, and sample is buffer plus your protein of interest. Then the photodetector is detecting, and then in the output you're getting the wavelength and the absorbance. From there you can actually get the concentration of the substance. Okay, now this is a very interesting concept. Try to see if this is new to you, or if this is not new to you, congratulations. Okay, but this is important and this is interesting. Let's have a look. So, hyperchromic shift. What happens here is often happens. So, hyperchromic means it is this wavelength. This is actually the absorption is increasing. Okay, so emission maxima, this is actually increasing. So, this happens when a molecule undergoes change in conformation, interacts with other molecules, or experiences a change in its environment that leads to an increase in the absorbance or fluorescence intensity. Okay, that is one thing. Then there is hypochromic. Hypochromic, it should definitely come down, right? So this happens when there is a reduction in absorbance or fluorescence intensity. This can result from the changes in the molecule's environment, such as a decrease in the solvent polarity or alteration in the molecular structure. Then there is red shift and blue shift. So what happens in the red shift? Right. So see, for example, a shift in absorption or emission towards shorter wavelength might occur due to an increase in the electron density or change in electronic structure of the molecule. This is called your hypsochromic or blue shift. And in case of the red shift, which is bathochromic shift, the shift is towards a longer wavelength, and that is often because of change in solvent polarity and interaction with other molecules and change in the molecular structure. Okay. So we will try to understand this better. So try to have a look at this. This is a question. So in the above kind of graph, this is how it was initially, and then it became this, right? It from A it went to B. So what kind of shift do you see? Right. Firstly, it is moving towards a smaller wavelength. So if it is going to a smaller wavelength, then it is definitely blue shift. Blue shift is your hypsochromic shift. And if it is increasing in wavelength, that means if it is absorbing more, so that means it is hyperchromic shift. So the right answer would be 1 and 3, which is hypsochromic and hyperchromic both. Fine. Now try to answer these questions. They are also related to spectroscopy of amino acids. We are nearly done with today's lecture and the assignment questions. Try to solve these two. Which of the following amino acid has the highest molar extinction coefficient value? Which one do you think would have the highest molar extinction coefficient value? I was talking about this in the B. Okay, phenylalanine. Okay. For the second question, okay, you're saying A. Okay, now let us find out which one is the right answer. So, which of the following is correct about the absorption spectra of amino acids? So, bathochromic shift occurs when polarity increases. Hypsochromic shift occurs when polarity increases. Now, these are the kind of questions which are very tricky. You will have to remember, because without that it would become very difficult to figure out, right? Hypsochromic shift occurs when hydrophobicity increases. Bathochromic shift occurs when hydrophobicity increases. See, let's try to have a look at this too. Can anyone answer? Otherwise, we'll look at the answers. Okay. This is very confusing, I know. Let's see. Okay. So, the right answer is tryptophan. And why is that so? Because tryptophan is actually giving you maximum absorbance. Okay. So, if tryptophan is present in your protein, then you're very lucky because you can very easily quantify the protein. So tryptophan has very high molar extinction coefficient because of its indole ring. Then there is tyrosine, and lastly it is phenylalanine. Okay, and for this one it is hypsochromic shift. It occurs when polarity increases. Why is that? Because conjugation is reduced and consequently the energy gap is increased. So conjugation means the interaction between your pi orbitals, right? That is reduced, and so what happens? The resulting blue shift happens and the band moves to shorter wavelength, right? That is your hypsochromic shift, and that happens when polarity increases. Okay. Now we will maybe see. Oh, very nice. You have actually answered. Very good. Congratulations. But okay, sorry. Okay, never mind. We will look at the next question then. We'll look at the next question. So this is a question on spectroscopy again. And yeah, this is the last question for our class today. A peptide containing 85 amino acid residues with molar extinction coefficient of such and such, and then having a molecular weight of 10.2 kDa if the peptide contains 4 tyrosines and the absorbance value of so and so, then calculate the number of tryptophan and the concentration of the protein solution. The extinction coefficient values for tyrosine and tryptophan are this and this. Okay, this is a very common question. These kind of questions, all these numerical questions, especially on the spectroscopy, these are very useful for your gate. Okay, the NAT questions come, numerical aptitude test, these comes, and here all these questions would be very important. So try to practice as many as you can. So see here what happens, the molar extinction coefficient, I told you it depends upon the number of the residues, number of aromatic amino acid residues present. So they have already said that the molar extinction coefficient of tyrosine and tryptophan are this and this. So how many tyrosines are there, that is also given. So 4 tyrosines. So how many tryptophans are there, that you can find out because absorption is given. And you know Beer-Lambert's law. So that is how you have to calculate and you have to find out. Right? Let's have a look at the answer. Okay. You guys are answering. That is very good. Let's look at the answer. Okay. Very good. The answer is A. Why is it so? Because you see epsilon is equal to number of the particular residue into molar extinction coefficient of the particular residue, and you sum it up for all the aromatic amino acids. And then they have said that absorbance is given. So absorbance is equal to epsilon into C into L. L is often taken as 1 centimeter. Okay, L is taken as 1 centimeter and C is already given. C is your, no, C is not given. Oh, net molar extinction coefficient is given, right? So from there you just calculate and you get it as 6. That is the number of tryptophan residues present, and concentration they have asked you to find. So you already know absorbance. Excuse me, you already know absorbance. You know the molar extinction coefficient now. So then what do? You just divide, and then that is 1 centimeter this side. It gets cancelled because the molar extinction coefficient already has centimeter as its unit, centimeter inverse, and so you get 1 into 10 to the power minus 5 molar, which is equal to 0.01 millimolar. Okay. Thank you for attending the class. If you have any questions, you can ask. So this was the first week of our course, Fundamentals of Protein Chemistry. and tell me if you liked it. Tell me if you want me to improve anything. Suggestions are always welcome. Ma'am, can you please explain second last question again? How increase in polarity decreases configuration, right? So if you see this, increase in polarity means one is becoming more negative and the other one is becoming more positive, right? And so because of that, the conjugation is reduced. Conjugation means overlap of the pi orbital, and consequently, the energy gap is actually increased. Because of that, the blue shift is occurring. So, blue shift is actually shift to shorter wavelength, and blue shift can be seen when this is often associated with changes that result in higher energy transitions, right? For example, a shift in absorption or emission towards shorter wavelength might occur due to an increase in electron density or change in the electronic structure of the molecule. So you can read more about this to understand this better, but mainly our hypsochromic shift is associated with this change in electron density and change in the electronic configuration in the interacting molecules. Okay, when can we get the videos in YouTube? Whenever I upload, I might be uploading either today or tomorrow or day after, and then it will be available. So NPTEL will be sending you a mail that the video and the PPT, these two have been updated, and then you can always go and access. Okay. And for the references, okay, I will add references here. So there will be books that you can refer to, there will be websites that you can refer to, and these will be very useful for you to understand the whole concept better. Okay, I will attach the links to those websites and the books, and also small exercises. Okay, those will be very useful for you. How can I take PDF? You will get the PDF. You will get this whole PPT. Okay. And that PPT you can actually download and you can study from there. The PPT will have all the links also, and you can access those links to get all the resources that are necessary for your course. Okay. Thank you so much. So if there are no more questions, we'll end the class. Could you explain some amino acids have different? How is it generated? How is the alphabetical code generated? For that, you will have to study amino acid nomenclature. Why they have given? Acha, is that what you asked? They have different alphabetical code. How is it generated? Yeah, for that you will actually have to refer to the nomenclature of amino acids. So the very simple amino acid alphabetical codes, the names, so we already know. Besides that, there are also B and X. These are also amino acid names that are being given. Okay. And then there is, I think X is for both aspartate and asparagine. Together they are called X. Okay. And then there is B. You can look that up. What exactly B is, I am not able to recall at this moment. So for that, you just have to know about the nomenclature of amino acids, and then you can definitely know. Because see, a lot of amino acids, they are starting with the same initials, right? For example, alanine, and then you have aspartate, and then you have asparagine, and so 3-letter codes are easier to distinguish, because single letter code would be too difficult. All would be A then, correct? And that is why you have different single and different 1-letter codes. What professor to take fundamental of protein chemistry? Professor Swagata Dasgupta from Department of Chemistry, IIT Kharagpur. She is the professor who takes your class. Okay, you can go to her lectures. You can always go through the lectures. Whatever she teaches and whatever assignment questions are there, I make the PPT based on that. Is it possible to calculate the Okay, I am not sure about that. You will have to read about this. I will also read about this. Okay, I will not be able to answer right now. manually in exam. I'm not sure about that. I'll have to read about it. This is your hydropathy index, right? Yeah, but then we'll have to actually read. Advantage of this course in future use. Okay, so in the future use, this course is very actually scoring, because a lot of questions for your entrance exams and all will come, right? So after this, if any of you are interested in going for higher education studies, let's say PhD, let's say you're going into some industry where they are working with proteins, they're working with biomolecules or any kind of protein ligand interactions and all of these, so there this is going to be of a lot of use. Okay, and also there are these, there can be so much, there can be protein engineering. I come from a lab of protein engineering. Then there can be protein based therapeutics. So in all of these things this is going to be useful, plus your this also medicine, what do you call it, pharma, what do you call it, pharmacogenomics and everything, right? There also this is going to be useful, because these are the very basics of biomolecules, and this finds applications in every field. So is histidine aromatic? Histidine is not actually classified as an aromatic amino acid, right? Why is that so? Because it is exactly not having this conjugate double bond structure. Look at histidine. Where is histidine? It's not here. Okay. Nah, histidine is not here, but histidine is, we saw a structure of histidine. Yeah. Yes, you see, this is not exactly conjugate double bond. This, there is resonance, but then this is not considered aromatic as such based on the conventional pharmacoinformatics. Okay, okay, great, thank you. Yeah, so this is very basics, right? So in basics find applications everywhere. So these are some of the potent future uses. You guys will grow up, you guys will do something great, I hope. So there also you can make use of all of these concepts and all if you want. So thank you everyone. If you don't have any more questions, we can end the session today. Okay. I'll see you again next Tuesday with week 2 lectures. Can we study all protein mutations? What is all protein mutations? Every amino acid can be mutated to 20 other amino acids. So that is like an infinite number of mutations are possible. But then you have to know which one you want to make, right? So exactly what mutation is going to give what effects. Some rationale should be there on your mind, and then you can design mutations and you can study the effect of those mutations on the functions and the properties of the proteins. You can design new proteins. Means disease oriented mutations. Yes, of course there are databases also, and now just by entering the sequence of an amino acid you can actually find out whether some kind of mutation is a missense mutation or not, and then what implications will it have in future, what kind of diseases can it be associated with, right? And there also all of these concepts will come, because a mutated amino acid means it will have different side chain properties. So different side chain properties means it will have in different, say, pi, different interactions with other amino acids. Because of that, maybe the protein protein interaction or protein DNA interaction is affected. Because of that, some havoc is going to happen. Okay. Thank you so much for your time. Thank you for being present with me all through the lecture. Okay. See you again. See you next week. Bye bye. let's say if you want to go for higher studies.